Hello everyone and welcome to the Picture Education. In this particular video we are going to talk about the buffers biochemistry, the lecture number 3. What are buffers? So buffers are the solution that resist changes in pH upon the addition of an acid or a base. They are extremely important in a biological system because they help in maintaining the blood pH and also help in maintaining the physiological pH inside the cells. So if we look at the standard definition of a buffer is a solution which contains a weak acid and its conjugate base or vice versa. For example, acetic acid, which behave as a weak acid, can dissociate into its conjugate base, that is acetate, and can release a proton moiety. Also, a buffer is a Le-Chatelier principle in action. So what is the Le-Chatelier principle? So Le-Chatelier principle says that there must be an equilibrium between the concentration of a reactant and the concentration of a product. So for a given particular buffer, the concentration of the acid and the concentration of its conjugate base must be equal, and since there is a equilibrium established between the buffer, and if we tend to destroy this equilibrium either by adding acid or by adding base, so what the system tend to do is to find the new equilibrium where it can adjust its concentration of product and the concentration of reactant to be in equilibrium. For example, if we take acetic acid and we add a base species into it, so what the system do is the acetic acid dissociate into its conjugate base and the proton is released. So the released proton react with the added base and it will form a water. To be in Le-Chatelier principle, the reaction must be reversible, so the conjugate base can react reversibly with the water and can form the acetic acid again, and the added base moiety is also reformed again. So this is how Le-Chatelier principle work for the buffers. So how buffering action of a buffer work? So at any given particular time we have an equal concentration of the acid and its base. So now if we add a acid into the system, so what a base do is base actually react with the added acid and it form a more of the weak acid. So after the addition of acid there is a formation of more of weak acid than its conjugate base, and on the contrary if we add a base into the system, what acid do is it dissociate into the proton and a conjugate base. So after reaction there is a formation of more of the conjugate base, and there is a less concentration of the acid. So even after the addition of the acid there is no net increase in the proton concentration, and even after the addition of the base species there was no net increase in the base concentration. So this is how the buffering action of a buffer works. And there is an equation called Henderson Hasselbalch equation which describes the behavior of weak acid in solution and allow us to calculate the concentration of a acid and a base at various pH. So we know that the acid can dissociate into a proton and a conjugate base. So we can write a dissociation constant for it. So K equals the concentration of proton into the concentration of the conjugate base by the concentration of the acid. And now if we take a log on both sides and rearrange the equation, so the equation comes the minus log of the proton concentration equals the minus log of the dissociation constant plus log of the conjugate base by its acid concentration. So we know the minus log of the hydrogen ion concentration is also known as pH, and the minus log of K is also known as pKa. So if we have to describe this equation in the term of pH, so the pH equals to pKa plus log of the conjugate base by its acid. So if the concentration of the base and the acid is equal, so at that particular concentration the log value becomes 0 and pH becomes the pKa or the dissociation constant. And there is a term called maximum buffering capacity, meaning how a buffer can work in effective buffering range. So a buffer is effective only in the pH range of plus or minus 1. So for example, to prove that, if we take a acetic acid where the concentration of acetic acid is way more than the concentration of its conjugate base, and if we add a NaOH solution to it, so after adding NaOH solution the free proton will react with the base and will form water, and after a particular point there will be no free proton in the solution. At that time, the acetic acid will dissociate into its the conjugate base and the proton, and the added NOH will react with that proton and will form water. So as we gradually increase the NOH or the base species, a particular point come where the concentration of acid becomes a concentration of the its conjugate base. So midpoint is the point where the concentration of acid and its conjugate base are equal. So at this point pH and pKa are both are equal. So now if we continue adding the add the OH or the base species, so what happens is the acetic acid lose more of its proton and the formation of a conjugate base is more and more, and there is a come equivalence point or the end point where a acetic acid lose all of its proton, and there is no, and it loses its buffering capacity. So the purple bar here representing the effective buffering range, where can the acetic acid actually buffer in very effective range. So some buffers are polyprotic in nature and they have more than one acidic proton. So the Henderson Hasselbalch equation only represent the removal of one proton from an acid and equilibrium between an acid and a conjugate base. But in a polyprotic acid, the all of the proton do not dissociate at the same pKa, but they are released sequentially at different pKa. So if we take a phosphoric acid and if we at the pK of 2.14, it releases one of its proton, and as we gradually increases the pH or the pKa, at 7.2 it releases another of its proton, and as we go higher and higher at the higher pK, which is at 12.4, it releases the last of its bound proton. So these are the polyprotic proton where they can release the proton at different pH or the pKa's. And there is also called a total buffering concentration. So total buffering concentration is the concentration of an acid plus the concentration of its conjugate base. For example, if a buffer is made of 0.1 molar acetic acid and a 0.1 molar of sodium acetate, also known as the acetate buffer, then the total concentration is just the addition of both, which is 0.2 molar concentration. I hope you liked the video and this was informative for you. If you like, please stay tuned in for the more videos, and thank you for your patient listening.